Hi, I'm Mike Kirkness. I'm president and CEO of 3Helix. I'm excited to be here today to tell you about what we've been doing for the past 2 years and where we're moving in the next 5. 3Helix is a company that's focused on collagen targeting, and we've been developing prognostics for fibrotic conditions. Currently, we're doing our Series A round. We're requesting $5 million. 3Helix is a platform diagnostic company. For the past 15 years, our founders have been researching collagen damage as a biomarker. And what that means for 3Helix is we have fantastic IP protection. We have exclusively licensed 2 compositional matter patents, one from University of Johns Hopkins and one from University of Utah. Additionally, we have a method patent exclusively licensed from Johns Hopkins and 2 provisional method patents that we have recently applied for this year as 3Helix. Currently, we sell an RUO product line. But moving forward, we'd like to move into the clinical histopathology space. We will be releasing an ASR towards the end of 2021 with a focus on NASH. Moving forward, though, we'd like to expand our reach from clinical histopathology into non-invasive techniques such as medical imaging and serum testing, and moving from NASH into multiple fibrotic conditions such as IPF, kidney fibrosis, AMD, and bone marrow fibrosis. Coming back to our main value proposition though, the liver fibrosis market is growing. The liver fibrosis market in 2015 for NASH was 16 million. It's predicted by 2030 to grow up to 27 million. Now, this is the progression for liver disease here based on non-alcoholic fatty liver. So we can see here a healthy liver moves into non-alcoholic fatty liver, moves into NASH, which is made up of 4 different stages, and then into cirrhosis. Now, not all patients will travel through this progression, and not all patients will even make it all the way through. Some patients will pause at non-alcoholic fatty liver, as we can see by the numbers here. Some patients will progress into NASH and stay at stage 1. Others will progress rapidly from fatty liver into cirrhosis, but we don't know which patients will move forward. At this point, there is no NASH therapeutics on the market. Intercept recently filed an NDA for their OCA drug, and this drug currently costs about $80,000. It's predicted to come down to 20 based on the size of the NASH market, however. So 16 million patients on the low end at $20,000 is $320 billion for annual cost for the medical insurance and the clinicians and patients. A huge cost that is going to be coming down the road that we need to learn how to deal with. So all of these patients, though, will not progress. I told you that NASH is a very heterogeneous and slow progressing disease. So we need to determine which patients need to be treated by these new therapeutics. So this infographic here shows us there's out of 100 patients with non-alcoholic fatty liver, 33% will move into fibrosis and only 20% will be rapid progressors. Now, this 20% actually has been suggested to be quite a bit lower in some very recent data, but it's still up to be seen whether that is convincing. However, we do know that a very small population of patients will be rapidly progressing. So it is very important to striate that patient population to determine who needs therapeutic intervention and who can just be diagnosed or treated with exercise and diet. So current biopsy methods to diagnose liver fibrosis are considered the gold standard. Some people will tell you that it's medical imaging. However, every new diagnostic gets benchmarked against biopsies. Thus, biopsies are still considered the gold standard for liver fibrosis. This is the current state right now, is Mason's trichrome highlighted in blue here. And Mason's trichrome is a true diagnostic. It does not tell you anything about a prognostic or prognosis in the future. It tells you a snapshot of the liver disease on that day. So it tells you you have stage 1 NASH today. It doesn't tell you what you have 2 weeks down the road or 6 months down the road, and whether you'll become cirrhotic in 6 months or whether you become cirrhotic in 10 years or whether you'll never become cirrhotic. And that's an important differentiation to make, is that we know what patients are now. We know they're in stage 2 NASH, but we don't know if they're prognostic. We don't know if we have to treat them, and we may be treating people that don't need treatment. And that's where we come in. So 3Helix is a collagen, has commercialized collagen hybridizing peptides, which is a very short peptide here, 9 nanometers, and collagen molecules 300 nanometers. So when our probe binds into damaged collagen here. So 2 alpha chains from the collagen, 1 CHP. It will reform the triple helix. It is not fixing it, it's targeting it. So our collagen molecule is targeting so we can visualize where the damage is occurring. What does that mean in terms of fibrosis? Okay, so fibrosis is an upregulation of collagen or an overgrowth of collagen, so a fibrotic scar. This doesn't mean that it's only fibroblasts are only producing collagen. It means that fibroblasts are producing collagen and MPs are actually upregulated at the same time. So now fibrotic scar is actually a very active state. So we can now see that we have collagen produced, collagen damaged, and then we have this activity going back and forth. As we have more collagen damage, that actually gives more feedback to the fibroblast to grow more collagen. And then we have this kind of snowball effect where we're growing more and more fibrotic scar. So CHPs allows you to visualize the activity of the fibrotic scar, not just the fibrotic scar itself or how much collagen there is. It's the damaged collagen that you can see. That's a very clear distinction from collagen 1 and 3 antibodies, which are visualizing total collagen. We are visualizing damaged collagen. We're visualizing the dynamic nature of the fibrotic scar. We can see that here in lung fibrosis model, bleomycin. At time point 0, no fibrosis. As fibrosis is progressing, we can see more and more activity, more and more damaged collagen. So we've used this as a prognostic tool. So we had access to wedge biopsy samples in here, and we had a subset of patients that had high level of CHP binding, and we had a subset of patients that had low level of CHP binding. We knew, because this was a few years ago, whether the patients needed a transplant or whether they did not need a transplant. And we were able to determine that patients that had high levels of CHP binding did need a transplant, and patients that had low levels of CHP binding did not need a transplant. So CHP fluorescent intensity was able to predict whether a patient needed a transplant or not. Interesting to note though, METAVIR staging did not correlate with the prognostic. So as I told you before, METAVIR staging is Mason's trichrome staging, and that is just a diagnostic. So this here is a mix of stage 3 and stage 4 fibrosis, and I think actually there was more stage 3 than there was stage 4, and it did not have any correlation between the two. Dr. Alistair Burt is currently working on this for NASH with paired biopsy samples in his biobank. So how do we want to come to market? We want to come to market as an analyte-specific reagent or an ASR. So currently we're selling a histopathology product line, and we're doing liver fibrosis testing with Alistair Burt. As we move into the ASR release, we chose this route for ASR because it's much less capital intensive, it's very quick to market, and it allows us to grow our market size by doing multiple fibrotic disease states. So this is at the end of 2021, and our liver fibrosis data should finish mid 2021 to late 2021. So we should be releasing a paper at the exact same time we're releasing our ASR data to encourage clinicians to develop laboratory tests based on our ASR product. As we finish off our liver testing, which we do need to expand to increase the clinical trial testing size, we will be moving into other fibrotic states such as kidney or lung fibrosis, which use biopsies in their diagnostic and prognostic pathways. To move into the European regulatory filing, though, we do have a much higher bar to meet, so it will be we will have to have more data, which is why we're moving into the larger biobanks as we release our ASR. At the same time, we are doing non-invasive development as well. We do have an ELISA kit that we have developed. We're still optimizing that, and we have started doing some medical imaging studies. So I just wanted to highlight our partners for how we're getting our liver fibrosis ASR to market is IQ Products. They're going to be our ASR register, and we're working with Newcastle Node and Dr. Alistair Burt. ARUP, we're doing a bone marrow study with, but also using liver fibrosis data to determine how to use it in clinical auto stainers. Further development for collagen as a biomarker, we're looking at non-invasive techniques. This is our ELISA kit, which is shown binding different collagen fragments at different rates. In this case here, we have a near IR imaging probe of lung fibrosis. We can see that we have a very bright spot on control. That's just our liver, and the CHPs have not been processed yet. However, if we just focus on the lung, we can see a high level of binding on the CHP in the lung here, low levels of binding in the control. And if we excise the lungs, we can see a high level of binding in the lung here and a very low level of binding in the lung for the control. As we cut up the liver or the lung here, we can take a look without any additional staining and see a very high level of collagen damage in the central part of the lung, which would be expected for our model of lung fibrosis. So our revenue is going to be driven by our ASR product. By 2024, we expect to be profitable at $14 million revenue a year. In 2023, this is our break-even point around the six-month mark. And again, we're still being driven by our ASR pathology line. In 2024, we have predicted a very conservative 7.5% for market penetration. I think we can easily increase this with our key opinion leaders that we have put in place. Our company has a very strong leadership team. We have Noah Nasser as our chairman of the board. He's done 4 or 5 biotech exits. He's currently fundraising for Ceramune, and he has decades of experience in the collagen diagnostic space, and he's a believer of our ASR product launch and has helped us put in contact in many different professional situations for us. Michael Fugman is a board member, and he's kind of been our supervising CFO. He's been a CFO for decades, and he's been helping us get our finances in order and getting ready for our venture capital round. Alistair Burt, again, world-leading pathologist, editor of McSween's Pathology of the Liver for the past 6 editions, current director of the molecular pathology node, one of the main pathologist for the invention of the ELF test, which is the very first non-invasive liver fibrotic diagnostic. And he's still very excited by our technology and the potential of it being a prognostic. Myself here, first-time CEO, and I've done multiple other startups in high-level, high-performance sport and biomechanics, but I have focused in on this biotech project because it is very exciting to me. I did my PhD in collagen mechanics and degradation, and I am fascinated by collagen degradation. So how are we going to use our proceeds for Series A? We are focused on growing our biobank access, growing our clinical study sizes, which will be the majority of the funds. We do need to move into a new laboratory space. So we have to develop our laboratory space, moving out of the incubator to allow for our team to grow from 4 to 15. In addition, we need to move our peptide production in-house so we can release our ASR product launch while maintaining our quality control insurances. So if anyone has any questions, I will be happy to take those now, and I'm excited to be here today. Thank you so much.